There's a particular kind of quiet that settles in when you stop moving. Not peace exactly, something closer to stillness. The kind where you become aware of your own breathing without meaning to. And in that stillness, if you are able to go small enough, smaller than the cell, smaller than the structures inside it, you would find something happening that has no pause button, no night shift, no rest. Something is always breaking. Welcome to the Sleepy Biologist. If you're here for the first time, I'm glad you found this, and if you've been here before, you already know what this is. This space exists genuinely because of the people who support it. Channel members, those who've sent super thanks, those who quietly hit like and never said anything, that matters more than I can say cleanly in a sentence. So I won't try to. Right now, as you lie there, wherever you are, whatever weight the blanket is putting on your legs, your DNA is being damaged. Not catastrophically, not in any way you'd feel, but the damage is real, and it is constant. And it has been constant since before you were born. The number is hard to hold. Each of your cells, and there are around 30 trillion of them, sustains somewhere between 10,000 and 1 million molecular lesions every single day. Some sources say more. What that means in plain terms is that the text of you is being smudged and torn and chemically altered over and over again every hour you exist. And you've never felt it once. That's the part that gets me, honestly. Not the damage, the silence of it. You feel a paper cut. You feel a headache building somewhere behind your left eye. But this continuous molecular unraveling happening in every tissue, every organ, every cell currently keeping you alive, nothing, not even a whisper of sensation reaches you. Part of what makes this possible, the damage I mean, is that DNA isn't inert. That's a thing people assume, that genetic material is locked away and stable, like something preserved in amber. But it isn't. It's a chemical. A long, complicated, astonishingly intricate chemical, yes, but still a chemical, and chemicals react. They react with water, with heat, with oxygen, with the byproducts of your own metabolism. They react with light. They react with each other. Even the warmth your body generates, the ordinary 37 degrees Celsius that keeps your enzymes working, is enough to shake loose certain bonds. There's a process called depurination. It's not dramatic. A purine base, adenine or guanine, 2 of the 4 letters that make up your genetic code, loses its grip on the sugar it's attached to and simply falls away spontaneously. No external trigger, just thermal noise and time. In each of your cells, this happens roughly 5,000 times a day. 5,000 in one cell per day. The hum of a refrigerator was going somewhere in the back of the building where I first read this, and I remember stopping, reading it again 5,000 times in one cell. There are 30 trillion cells. The math doesn't really help. What helps slightly is knowing that this isn't a modern problem. This isn't something that started when the atmosphere changed, or when humans invented things that emit radiation. This damage has been happening for as long as life has existed. The first cells on this planet dealt with it. Bacteria deal with it. Every organism that survived long enough to reproduce has, somewhere in its biology, something that handles this. Something that runs constantly without instruction from you, correcting what the chemistry undoes. But that comes later. For now it's enough to just sit with the fact of it. Your DNA, right now, in this moment, while your breathing slows and the room does whatever quiet rooms do at this hour, is being altered. Some bases are losing their footing. Some chemical bonds are bending under the weight of oxygen byproducts. Somewhere in your skin cells, which are still cycling and dividing even as you rest, a strand has just broken, and something is already moving toward it. You don't have to do anything. You haven't had to do anything about this, not once in your entire life. There's a whole infrastructure inside you built for exactly this. Ancient, reliable, running so far below the level of conscious experience that sleep doesn't interrupt it. It runs in the dark just fine. Water is the thing we reach for when we talk about life. It shows up in every origin story, every definition of habitability, every checklist scientists use when they were looking for something alive somewhere else. And there's good reason for that. Water is where chemistry happens. It's the medium, the solvent, the condition under which molecules find each other and do what molecules do. But water is not passive, and inside a cell it is not kind. This is the part that took me a while to really settle with. Not intellectually, but in some quieter way. The same molecule that makes life possible is also continuously taking it apart. Not maliciously, not with any direction at all, just chemically. Just because that's what water does when it's warm and close to other molecules and has nowhere else to be. The process has a name, hydrolysis, from the Greek and water and loosening. And what it loosens, among other things, are the bonds holding your DNA bases to the sugar-phosphate backbone that gives the double helix its shape. Purines, adenine and guanine are particularly susceptible. The bond connecting them to deoxyribose is a glycosidic bond, and it hydrolyzes spontaneously, without prompting. At body temperature, in the ordinary wet interior of a living cell, it simply lets go. What's left behind is called an abasic site, or an AP site, apurinic. When the purine is the one that's gone, the backbone is still there, the strand is still intact. But where a letter used to be, there's now a gap in the sequence, a missing instruction, a place where the code has gone quiet. Around 5,000 of these happen per cell per day. I was sitting outside when I first wrote those words down, and there was a tap dripping somewhere nearby, just faintly, one of those sounds you stop hearing until you hear it again, and the number felt strange in the context of that sound. 5,000 in one cell from water, from warmth. And it doesn't stop there. Water isn't the only hydrolytic agent doing this work. There's also deamination, a different reaction, slightly different chemistry. Cytosine, one of the 4 DNA bases, loses an amino group. What it becomes is uracil, and uracil doesn't belong in DNA, it belongs in RNA. So now there is a letter in your genetic text that is not wrong exactly, but misplaced. A letter from a different alphabet sitting in a sentence it has no business being in. This happens roughly 100 times a day. Per cell, 100 times. Quietly, without any signal you'd feel. There's something I find myself returning to when I think about this, which is that none of this is the consequence of damage in the way we usually mean damage. No trauma, no toxin, no radiation, just life. Just the ordinary chemical reality of being a warm, wet organism with a genome that must be maintained inside conditions that are, at the molecular level, relentlessly erosive. The warmth that makes your enzymes work is also the warmth that shakes bonds loose. The water that makes cellular chemistry possible is also the water that hydrolyzes your bases away. Your existence is, in some technical sense, in constant low-grade conflict with its own conditions. That sounds more dramatic than it is, because the other side of it, the part that doesn't get named as often, is that cells have been handling this for somewhere close to 4 billion years. Not 4 million, 4 billion. The first cells on this planet almost certainly face the same hydrolytic pressure that yours do tonight. And they developed, through whatever processes evolution runs on, mostly failure, mostly death, slowly occasionally survival, a set of responses. Repair pathways, enzymes that scan for missing letters, proteins that recognize the particular shape of an abasic site, and know, from that shape alone, that something needs to be corrected. The water doesn't win, or rather, it wins constantly and something constantly wins it back. What that something is, how it moves, what it recognizes and what it misses, that's where this is going. But there's no urgency in getting there. The repair systems have been running since before you fell asleep. They'll still be running when you wake up. The AP sites forming in your cells right now in the quiet hours are already being found. Somewhere inside you an enzyme is sliding along a strand of DNA. It has already read 1,000 times tonight, checking shapes, checking fit, pausing at the place where something is missing. It doesn't hurry, it doesn't need to. You've felt it on your face. That particular warmth. Not heat exactly, more like pressure. The way light in the afternoon has weight to it. You turn toward it without thinking, something in you has always done that. But light doesn't stop at the surface. UV radiation, the wavelengths just beyond what your eyes can register, passes through the outer layers of skin and reaches the cells still dividing underneath. Most of it is absorbed. Some of it isn't absorbed cleanly. When a UV photon collides with a DNA strand in one of those cells, something specific can happen. Two pyrimidine bases, thymine and thymine, or sometimes thymine and cytosine, sitting right next to each other along the same strand, or can form a bond that shouldn't exist. A covalent bond, directly between them. The two bases lock together into what's called a cyclobutane pyrimidine dimer. A thymine dimer, in common shorthand, is a small thing geometrically, just 2 letters in a 3 billion letter sequence fused, but the double helix buckles at that spot. The structure of DNA depends on its bases pairing across the strand, adenine to thymine, cytosine to guanine, and that pairing depends on geometry. The right angles, the right distances, the right shapes facing each other across the helix. Dimmer distorts that. It introduces a kink, a wrong angle, a place where the regular architecture of the molecule fails. I keep thinking about origami when I think about this. The way one folds in the wrong direction, just one, changes what the whole thing becomes. But that's probably not the right comparison, and I'm going to let it go. The DNA doesn't fold like paper. It does something stranger and more specific. What it does is this. The kink gets noticed. Not by anything with awareness, not by a cell deciding to look. A protein, part of a larger repair complex, moves along the DNA and encounters the distortion. Its binding site is shaped for normal helix geometry, and when the helix isn't normal, the fit changes. That change in fit is, in its own molecular way, a signal. The protein stalls. Other proteins arrive. This is nucleotide excision repair, and it is unhurried. The complex that assembles at the site of a thymine dimer is made of multiple proteins. One unwinds the helix slightly. 2 endonucleases cut the damaged strand on either side of the lesion. One cut upstream, one cut downstream, and a short stretch of DNA, somewhere between 25 and 30 nucleotides long, is lifted out, just removed. The damaged section taken cleanly away. What's left is a gap, a single-stranded gap in the DNA, with the intact complementary strand still present on the other side. And that intact strand becomes the template. A DNA polymerase arrives, reads the undamaged sequence across from the gap, and fills it in, letter by letter, correctly. Then Ligess seals the final nick, and the kink is gone. The whole process, recognition, assembly, excision, synthesis, ligation, happens in minutes. Some sources say faster. I find that genuinely hard to picture, which I think means the scale is right. Here's the thing that sits strangely with me about thymine dimers specifically. There's an enzyme called photolase that repairs them directly, uses the energy from visible light to break the fused bond, reverting the bases to normal without cutting anything out. Bacteria have it, many animals have it, fish have it, birds almost certainly have it, humans don't. Somewhere in our evolutionary past we lost the gene, or the gene drifted into uselessness and was never selected back. So while other animals can, in a fairly direct sense, use light to undo what light did, we can't. We use nucleotide excision repair instead. The slower, more complicated pathway. The one that cuts the damage out entirely rather than reversing it. Why? There isn't a clean answer. There's no story about why exactly placental mammals lost photolase activity. It just happened. And what we have instead works well enough that the loss didn't kill us. Well enough being a relative term, because NER isn't perfect, it's prioritized. Actively transcribed genes get repaired faster than silent ones, which makes sense but means the rest of the genome accumulates damage longer. And when UV exposure is high and sustained, the system gets overloaded. Lesions accumulate faster than they're cleared. There is a condition called xeroderma pigmentosum. It's rare. People born with it have mutations in one or more of the proteins required for nucleotide excision repair. Without functional NER, UV-induced thymine dimers go unrepaired. They accumulate. The risk of skin cancer in these individuals is elevated by something like 10,000-fold compared to the general population. They cannot go into sunlight without substantial protection. That number, 10,000-fold, is what a functioning NER pathway is silently doing for you every time you step outside. You don't thank it. You don't know it's working. You feel the warmth on your face and turn toward it and come inside later and lie down, and the repair is already hours underway, finding the kinks, cutting them out, filling the gaps, sealing the strand back together. Outside right now, the UV index is 0. Middle of the night, no new dimers forming. And somewhere in your skin, the ones from earlier today are still being found. There is something that moves along your DNA right now that has no eyes, no awareness, no sense of purpose in any way you could recognize as purpose. And yet it finds what is broken with a reliability that most human systems never achieve. It's called a DNA glycosylase. And before anything else about it can be said, I think it deserves a moment of just attention. Not reverence exactly, more like the feeling you get when you watch someone do a very small thing with complete competence. The kind of thing that would be invisible if it weren't so quietly perfect. A glycosylase is a protein, an enzyme. Its job, the only job it has ever had, is to move along a strand of DNA and find the bases that don't belong. Not by reading the sequence. It doesn't read. By shape, by fit. By the way, a damaged base occupies space differently than a healthy one. And the way a protein shaped over millions of years of evolutionary pressure can feel that difference in the time it takes to make and break a single molecular contact. The air in the room right now has a particular stillness to it, the kind that happens around midnight, when even the sounds outside have thinned out. And I find that it helps, somehow, to think about glycosylases in that kind of quiet, because what they do is quiet, thoroughgoing, unannounced. Here is what actually happens as best we understand it. The glycosylase doesn't just slide passively along the helix. It interacts with it. At intervals, it does something called base flipping, reaching into the helix and pushing a base out of its normal stacked position, rotating it outward into the enzyme's active site. For a normal undamaged base, this produces a poor fit, and the base flips back. For a damaged one, the fit is better, and the enzyme holds it. That holding triggers the next step. The glycosidic bond, the chemical connection between the base and the deoxyribose sugar it's attached to, is cleaved, hydrolyzed. The base is removed. What remains is an abasic site, an AP site, the same kind of site that forms spontaneously from depurination, which was the subject of the last chapter. Only now it was created deliberately as a repair intermediate. The damage is gone. What's left is a gap in the information, a missing letter, a silence where a base used to be. And that silence is the signal for everything that follows. An AP endonuclease arrives. It recognizes the abasic site by its shape. The missing base changes the geometry of the backbone subtly but detectably, and it cuts the DNA strand on the 5' side of the gap. Just one nick. Precise. Then a phosphodiesterase trims the leftover deoxyribose phosphate fragment that base excision repair leaves behind. Then DNA polymerase beta, a small specialized polymerase used almost exclusively for repair rather than replication, fills the single nucleotide gap using the intact complementary strand as a template. One correct base, insert, then DNA ligase seals the nick in the backbone. Done. The whole pathway, glycosylase, AP endonuclease, phosphodiesterase, polymerase beta ligase, moves through a damaged site in what amounts to seconds of biological time. For a single nucleotide repair, the patch is exactly one base long. For longer patches, polymerase beta sometimes extends a few bases beyond the original site, and a different set of enzymes handles the cleanup. I want to pause here for a second because I think it's easy to read that sequence. The list of enzymes arriving in order, each doing its specific thing, and feel like you understand it. And you do, in one sense. But I find it genuinely hard to hold the spatial reality of it. These are proteins. They are nanometers in DNA. The DNA strand they're working on is 2 nanometers wide. The base being removed, the gap being filled, the nick being sealed. All of this is happening at a scale so far below anything your nervous system has ever perceived that I'm not sure the word small is adequate anymore. It's not small, it's a different order of existence entirely, and it's happening right now in every tissue, continuously. What I find most interesting, and this is maybe a personal bias, is the specificity. There are multiple different glycosylases, and each one is tuned to a particular type of damage. Uracil D-N. Glycosylase finds uracil in DNA, the base that forms when cytosine spontaneously deaminates, the wrong alphabet letter that doesn't belong. OGG1, 8-oxyguanine DNA. Glycosylase 1 finds 8-oxyguanine, the oxidized form of guanine produced when reactive oxygen species from your own metabolism collide with your DNA. MUTYH finds adenine that has been misincorporated across from 8-oxyguanine before OGG1 could remove it. Each enzyme, each specific lesion, each shaped by the particular problem it evolved to solve. The system is not a single tool. It is a long accumulated collection of very targeted responses, assembled across evolutionary time, each one added because the damage it addressed was common enough and dangerous enough that cells without the solution died more often than cells with it. But here's what I want to be honest about, because I think it matters. This system is not perfect. Glycosylases miss lesions. AP sites that go unrepaired too long collapse into strand breaks. Polymerase beta, for all its reliability, introduces errors at a low but non-zero rate. The backup systems that catch those errors sometimes miss too. There is no level at which the repair is absolute. What there is, what you have right now running without a single instruction from you, is a system so deeply layered, so redundant in its structure, so shaped by the weight of billions of years of selective pressure that most of the damage gets caught, most of the lesions get fixed, most of the abasic sites get filled before they become something worse. Most. And in the space that word leaves wound, in the small percentage of lesions that slip through, that accumulate, that change the sequence in ways the downstream systems don't catch, there is aging. There is the slow drift of the genome over a lifetime. There is occasionally the first step towards something more serious. But not tonight, in all likelihood. Tonight the glycosylases is doing what they always do, moving along the strand, flipping bases, finding the wrong shapes and removing them with the quiet, unhurried accuracy of something that has never needed to hurry, because it was built for exactly this and nothing else in the only environment it has ever known. The nick gets sealed. The strand is whole again. Something further along the helix is already waiting to be found. Every cell that divides does something that should, if you look at it plainly, seem catastrophic. Both strands of the DNA helix, not one both, are severed, at the same time, completely. A double-strand break. No intact template remaining on either side of the cut. Just two blunt ends floating in the nucleus, separated from each other, surrounded by the proteins that will either fix this or begin the process of ending the cell entirely. This isn't rare. It happens between 10 and 50 times per cell cycle in somatic replicating cells. Per cycle. In cells that divide regularly, gut lining, skin, bone marrow, that cycle runs continuously. 10 to 50 times, both strands cut through. I keep coming back to this number. I've read it in multiple places now from different sources, and each time there's a moment where something in me wants to push back on it. Wants there to be a footnote that says, but most of these are very minor, or the majority resolve immediately. And there are footnotes like that in a sense. Most double-strand breaks are repaired before any consequence accumulates. But the break itself is real. The severance is real. The situation the cell finds itself in 10 to 50 times per cycle is genuinely one of the most dangerous things that can happen to a genome. A double-strand break left unrepaired can mean chromosomal rearrangement, loss of genetic sequence, fusion of chromosome ends that shouldn't touch. Any of these in the wrong gene at the wrong time can be the beginning of something the cell won't recover from cleanly. So the question isn't whether this is serious. It is. The question is how a cell handles something this serious this often with the accuracy it does. The fan in the corner of the room just changed pitch slightly, the way fans do when the temperature drops. And I noticed it without meaning to. Some part of the brain always cataloguing. There are two main pathways for repairing double-strand breaks. They work differently, they're used in different circumstances, and they have different error rates. The first is non-homologous end joining, NHEJ. It does more or less what it sounds like. The broken ends are detected, captured by a protein complex and ligated back together, directly, without a template. The Ku protein, a heterodimer, two subunits that form a ring beneath, loads onto each broken end and holds it. A kinase called DNAPKCS is recruited. The ends are processed, sometimes trimmed, sometimes filled, and then joined. It is fast, it is not always precise. NHEJ can introduce small insertions or deletions at the repair site. A few bases added or removed in the process of joining two ends that don't fit together perfectly. In most cases, if the break happened outside a critical gene, this doesn't matter. In some cases it does. NHEJ accepts a small error rate as the cost of speed. The second pathway is homologous recombination, slower, more accurate. requires the cell to be in a specific phase of the cell cycle, late S phase or G2, after the DNA has been replicated and a sister chromatid is available as a template. And this is the part that stopped me when I first properly understood it. The cell searches its own genome for a sequence matching the region that just broke. Not a rough match, an exact match, or near enough to exact that the repair machinery can use it as a template to reconstruct the missing sequence across the break letter by letter before the two ends are finally joined. That search happens across 3 billion base pairs, in a nucleus roughly 6 micrometers wide, in minutes. I don't have a comparison for this that I trust. I started to think of something about searching a city for a single address, and stopped because it didn't capture it. The scale is wrong. The mechanism is wrong. What the cell does is stranger and more specific than any spatial analogy I can build for it. It finds the match, usually, not always. There are breaks that don't get repaired correctly, breaks that get misjoined, ends from 2 different chromosomes that are captured together and fused when they shouldn't be. These events are rare in absolute terms and far more common in cells under sustained genotoxic stress. Cells exposed to radiation, to certain chemicals, to conditions that create more breaks than the repair machinery can comfortably handle. But in an ordinary cell, on an ordinary night, the breaks get fixed. 10 to 50 times per cycle. Fixed! And the cell continues carrying its repaired genome forward into the next division, where, somewhere in the replication process, or in the chemistry of just being alive, something will break again. There's a particular quality to the way certain biological facts land differently the second time you encounter them. The first time, you process them intellectually. You file them somewhere, nod at the number, move on. The second time, something shifts. The number stops being a number and becomes a situation. A real physical situation happening inside actual matter, in the dark interior of a cell that doesn't know you're thinking about it. The number in this case is 3 billion. That is roughly how many base pairs make up the human genome. 3 billion letters in a sequence that, stretched fully out, would run somewhere close to 2 meters long, coiled and packed and organized with extraordinary density into a nucleus that fits comfortably inside a space too small to see without a lens. And somewhere in that coiled, compressed, chemically active space, after a double-strand break has been detected and the broken ends have been secured by the Q complex, a search begins. The cell needs to find a matching sequence, not a similar one, not a rough neighborhood. The repair machinery requires a sequence that corresponds closely enough to the region flanking the break that it can use the intact copy as a template, base by base, to reconstruct what's missing, and it looks for that match by moving through the genome. Through all of it, or enough of it that the probability of finding the right stretch climbs toward certainty. This process is mediated largely by a protein called RAD51. It's a recombinase, a class of protein with an ancient evolutionary history, present in organisms ranging from bacteria to humans in various forms, which tells you something about how long this particular problem has existed and how persistently life has needed to solve it. RAD51 coats the single-stranded DNA at the end of the break, forming a filament, and that filament begins to probe the surrounding genome for homology. The word homology, here, means sameness, sequence sameness, and finding it requires something close to what I can only call, though the comparison isn't quite right, a continuous act of recognition. The RAD51 filament makes transient contacts with double-stranded DNA elsewhere in the nucleus. It touches a stretch, assesses the match, moves on if it's wrong, moves on and touches another and another. 3 billion base pairs of possibilities. I was rereading my notes on this late in the evening last week, and outside the window there was wind moving through something, a tree or a bush I couldn't see, and the sound of it was the kind of continuous white noise that makes thinking feel easier. I don't know why I remember that. Something about the rhythm of it felt right for this. The search takes minutes. And I want to be careful here, because it's tempting to frame this as miraculous. The word miracle has a way of short-circuiting genuine understanding. But what it actually is more interesting than miracle. What it is the outcome of selection pressure acting on a protein structure over hundreds of millions of years, until the protein's shape, its charge distribution, its dynamic behavior in the nucleus, produce something capable of this, not magic, accumulated fitness, the difference between a miracle and a very, very long optimization. RAD51 finds the homologous sequence, nearly always. When it does, the broken strand invades the intact double helix at that location, physically inserts itself between the two strands of the matched region, displacing one strand and pairing with the other. This forms a structure called a displacement loop, a D-loop, where the broken strand is now hydrogen bonded to its match, using it as a guide. DNA polymerase extends from that point, synthesizing new sequence across the break using the intact template. Letter by letter, correct letter by correct letter. And here is where the cognitive vertigo hits me every time. What is being written in these new bases is not new information, it is recovered information. The sequence was already there, already existing in the cell's other copy. The polymerase is not inventing. It is reading one strand and transcribing it onto another, rebuilding a stretch of genome that was severed and is now, base by base, being made whole again. After synthesis, the D-loop is resolved. The strands are separated, the ends are processed, ligase seals the remaining nicks, the break is gone, the sequence is restored. Not approximately restored. Restored. The accuracy of homologous recombination is high enough that, under normal conditions, it is considered essentially error-free. This is why cells prefer it over non-homologous end joining when the option is available. When the sister chromatid is present, when the cell is in the right phase of its cycle. Given the choice between fast and imprecise and slow and accurate, the cell, when it can afford the time, chooses accuracy. That preference, that biological tilt toward correctness over speed when the stakes are high enough, feels like something worth sitting with for a moment. It's not a decision. The cell doesn't deliberate. But the behavior is real. The outcome is real. And the outcome tonight, in the cells of your body that are currently replicating or have recently replicated, is that double-strand breaks are being found, and the genome on either side of each break is being read, and the correct sequence is being rebuilt. Somewhere inside you right now, 3 billion base pairs, and somewhere in there, a filament of RAD51 has just found what it was looking for. The strand goes in, the gap is read, the letters come back one by one, quiet and correct, into the place where they were always supposed to be. DNA replication is fast, remarkably stubbornly fast. In human cells, the replication machinery copies around 50 base pairs per second at each active replication fork, and there are thousands of forks running simultaneously across the genome during S phase. The whole 3 billion base pair genome, duplicated in a matter of hours. At that speed, errors happen, not many. DNA polymerase, the enzyme doing the copying, has a built-in proofreading function. As it synthesizes each new base, it checks the fit before moving on. If the fit is wrong, if a G has been placed across from a T instead of a C, or an A across from a C instead of a T, the polymerase can reverse, excise the misincorporated base and try again. This proofreading happens in real time during synthesis, and it catches the vast majority of errors. But not all of them. Some mismatches slip through. The fit is wrong but not wrong enough to trigger the proofreading function, or the polymerase moves on before the check completes. What results is a newly synthesized strand with a base in the wrong position, a subtle distortion in the helix, a GT pair, or a TA pair, where Watson-Crick geometry says there should be something else. This is what mismatch repair was built for. The core proteins in human mismatch repair, MSH2, MSH6, MLH1, PMS2 among others, scan newly replicated DNA for exactly this kind of subtle wrongness. Not the bulky distortions that nucleotide excision repair handles, not the missing bases that base excision repair addresses, just the quiet geometric imprecision of a misincorporated nucleotide sitting in an otherwise intact strand, disguised as normal DNA, except for the angle of its hydrogen bonds. There's something almost tedious about this task, in the best possible sense. I keep thinking about quality control in contexts where everything looks nearly identical, and only very close attention separates a pass from a fail. The mismatch repair proteins do this continuously for a genome 3 billion bases long, at a speed that keeps pace with replication. The light in the room had shifted slightly by the time I got to this part of my notes. Cloud cover maybe, or just the hour. And I noticed it the way you notice things when you've been sitting still for too long. The search itself is one thing, finding the mismatch is one thing. But mismatch repair faces a problem that has always struck me as genuinely interesting. The kind of problem that once you see it, you can't unsee. When the repair machinery finds a mismatched base pair, it must remove the wrong base and replace it with the correct one. But both strands are still present. Both strands contain a base at that position. One is correct, the template strand, the original, and one is wrong, the newly synthesized strand with the misincorporated nucleotide. How does the repair machinery know which is which? The sequence at a mismatch site doesn't tell you. Both bases are real DNA bases. Both are chemically legitimate. The mismatch itself only tells you that one of them is wrong, not which one. The answer, in bacteria, involves methylation. Bacterial DNA is methylated at specific sequence motifs shortly after replication, but the new strand lags behind. It's unmethylated for a brief window after synthesis. Mismatch repair proteins exploit this window, using the methylation status to distinguish old strand from new. The unmethylated strand is the new one. The new one is the one that might carry the error. Remove from there. In humans, the mechanism is less fully understood. There is evidence that the replication machinery itself leaves signals, nicks, gaps, associations with the replication factors that help mismatch repair proteins identify the newly synthesized strand. The details are still being worked out, which is worth saying plainly. This is an active area of research, and the picture is not complete. What is clear is that it works. The error rate of DNA polymerase alone, without proofreading, sits around one mistake per 100,000 bases copied. With polymerase proofreading, that drops to roughly 1 per 10 million. Add functional mismatch repair, and the final rate falls to approximately 1 error per billion bases. 1 per billion. To put a slightly different frame on that, if you copied a text 3 billion characters long and made 3 errors, you would have matched the fidelity of human DNA replication. 3 errors across a document that printed in standard font would fill a library. This is what mismatch repair contributes to that number. Not all of it. Polymerase proofreading does the heavy early work. But the final order of magnitude reduction, the difference between 1 error per 10 million and 1 per billion, is largely mismatch repair's doing. and when mismatch repair fails, when the proteins are mutated or silenced, the error rate climbs back up. Mutations accumulate faster, the genome drifts. There are hereditary cancer syndromes linked specifically to defects in mismatch repair genes. Lynch syndrome, associated with elevated risk of colorectal cancer and several others, is caused by germline mutations in MESH2, MLH1, and related proteins. The cancers that arise in these contexts are characterized by something called microsatellite instability, a pattern of mutation accumulation visible in short repetitive DNA sequences, a kind of genomic fingerprint of repair failure, which means the absence of mismatch repair is, in some cases, diagnosable, observable. The repair system leaves traces of its work, and its absence leaves traces of a different kind. Tonight, in the cells that have divided while you've been lying here, new strands have been synthesized. Mismatches, rare, but present, have been scanned for and found. The repair proteins have identified the newer strand, located the wrong base, cut it out along with a short surrounding stretch, and filled the gap with the correct sequence. Ligase sealed it. The error rate is approximately 1 per billion. You will not feel any of this. The strand runs on unchanged in any way you'd recognize, carrying the sequence forward. Right now, without any bad decision on your part, your cells are burning, and not dramatically. Nothing is on fire in any sense you'd recognize. But the mitochondria in your cells, those small membrane-wrapped structures distributed through the cytoplasm in numbers that vary by cell type, more in the heart muscle, fewer in fat, are oxidizing glucose, breaking it apart, extracting energy from the chemical bonds. This is respiration, and it is how you stay warm, how your neurons fire, how every protein in your body gets made and maintained and eventually replaced. This is also continuously producing something you didn't ask for. Reactive oxygen species. The phrase is clinical and slightly cold for what these things are, which is the metabolic exhaust of being alive. Superoxide hydrogen peroxide hydroxyl radicals, molecules carrying unpaired electrons, chemically unstable in a way that makes them grab at whatever is nearby. Proteins, lipids, cell membranes, and DNA. There's a smell in the room tonight, something faint, old paper maybe, or the particular dryness of a heater running, and I keep noticing it at odd intervals, the way you notice a sound that's always been there. When a hydroxyl radical collides with guanine, one of the 4 DNA bases, the one that pairs with cytosine, it can add an oxygen atom to the C8 position of the guanine ring. What forms is called 8-oxoguanine, or 8-oxo-G, depending on which paper you're reading. It's a small chemical modification. The base is still there, still attached to the backbone. It hasn't been removed. It hasn't broken the strand. To a casual inspection, it barely looks wrong, but it pairs wrong. Normal guanine pairs with cytosine. 8-oxoguanine, because of the extra oxygen at its C8 position, can pair with adenine. Specifically with adenine, in a configuration called a syn conformation, that is geometrically stable enough to fool the replication machinery. If a replication fork passes over an unrepaired 8-oxo-G before it's been removed, DNA polymerase may insert an adenine across from it instead of a cytosine. Now the mutation is real. What was a modified base, still correctable, still removable, has become a permanent change in the sequence. A GC base pair has become in the daughter strand, a TA base pair. One letter changed. In most locations irrelevant. In some locations not. The enzyme responsible for catching 8-oxo-G before this happens is called OGG1. 8-oxoguanine DNA glycosylase 1. It is a base excision repair enzyme, the same pathway described in the earlier chapter, and it is specialized for this specific lesion. OGG1 moves along the DNA and finds 8-oxo-G by the same base-flipping mechanism other glycosylases use. The modified base fits differently in the active site, and that different fit triggers cleavage of the glycosidic bond. The damaged base is removed, and a basic site is left. The downstream BER pathway fills the gap with a correct cytosine. The lesion is corrected. The sequence is preserved. But here is the thing I keep returning to when I think about oxidative DNA damage, the thing that makes it feel different from UV damage or hydrolysis or replication errors. Those sources of damage are, in some sense, external conditions, or at least separable from the core machinery of the cell. UV comes from outside. Hydrolysis is water acting on a vulnerable molecule. Replication errors are the cost of copying. Oxidative damage is different. It comes from the process that makes everything else possible. You breathe in, oxygen is delivered to your mitochondria, and your mitochondria do what they evolved to do, produce ATP, the energy currency of the cell, through a process that involves electron transport chains and carefully managed reduction reactions. And in that process, despite the extraordinary precision of the proteins involved, a small percentage of electrons leak. They react with molecular oxygen before they complete the transport chain. They produce superoxide. Superoxide becomes hydrogen peroxide. Hydrogen peroxide, in the presence of certain metal ions, becomes the hydroxyl radical. The same metabolic process that powers every contraction of your heart also produces molecules that oxidize your DNA. There is no version of aerobic life that separates these two things. The damage is not a malfunction, it is a side effect of a system that works. The mitochondria are not failing when they produce reactive oxygen species, and they are running, and the ROS are what running costs. Estimates of oxidative DNA damage vary by study and methodology, but the commonly cited figure, drawn from work going back to research by Bruce Ames and others in the 1980s and 90s, is around 10,000 oxidative lesions per cell per day. Most are caught by OGG1 and the BER pathway. Most are cleared before a replication fork arrives. Most leave no permanent mark, but OGG1 can be overwhelmed. Under conditions of elevated oxidative stress, illness, sustained inflammation, exposure to certain environmental toxins, even intense prolonged exercise, the rate of 8-oxo-G formation can outpace the repair rate. Lesions accumulate. The probability of a replication fork encountering an unrepaired 8-oxo-G increases. There is a backup system, MUTYH, another glycosylase, which recognizes adenine misincorporated across from 8-oxo-G and removes it, giving the cell a second chance to correct the situation. But MUTYH is downstream of the first error. It catches what OGG1 missed, after the mismatch has already been made. 2 layers of defense for one modified base. Both running right now in your cells because 10,000 times a day in each one is frequent enough that redundancy isn't a luxury. It's the only thing that keeps the error rate manageable. Your breathing slows a little when you're this close to sleep. The demand on your mitochondria drops slightly. Less activity, less oxygen consumed, fractionally fewer reactive species produced. It's not a large effect. The damage doesn't stop, but the rate at rest is lower than it would be if you were moving. There is something almost restful about that as a biological fact. Something in the rate right now is lower than it was an hour ago. Something is still running, catching what it can, quiet, continuous, already halfway through tonight's work. Not every break gets fixed in time. Not every lesion is found before the replication machinery arrives. Not every damaged base is removed cleanly before something downstream depends on it. The repair systems in your cells are extraordinarily capable. This entire script has been, in some sense, a description of how capable they are. But they are not infinite. They run at a rate. They have a throughput. And when damage accumulates faster than that throughput allows, something else has to happen. The cell stops. Not permanently, not immediately, not with any finality, just stops. The cell cycle, which drives cells forward from growth through replication through division, has checkpoints built into its structure at several points. The G1 checkpoint before DNA replication begins, the S-phase checkpoint during replication, the G2 checkpoint after replication and before division. Each one is a place where the cell cycle can be halted if the machinery detects something unresolved. The central sensor at most of these checkpoints is a protein kinase, ATM for double-strand breaks, ATR for stalled replication forks and single-stranded DNA. When either of these is activated by the damage signals, they initiate a phosphorylation cascade. One signal becomes many. The cascade reaches proteins that regulate the cell cycle engine, cyclin-dependent kinases, CDKs, and inhibits them. The engine slows, then stops. This is the checkpoint. A held breath, in biological terms. The cell neither progressing nor retreating, just paused, at a threshold, while the repair machinery works. How long the pause lasts depends on what's being repaired and how quickly it's resolved. For minor damage, the pause is brief. The repair completes, the inhibitory signals are reversed, the CDKs become active again, and the cell continues into the next phase. For more extensive damage, the pause extends, the checkpoint holds, the repair machinery keeps working, and the checkpoint sensors keep checking, and the cycle remains arrested until either the damage is resolved or a different signal takes over. There is a cold draft coming from somewhere, not unpleasant, just present, the kind that appears in old buildings at night when the temperature outside drops below what the walls can fully absorb. The different signal, when it comes, is apoptosis, programmed cell death. A word that sounds clinical and final and is both, but is also, and this framing matters, a protective mechanism. Apoptosis is not the cell failing, it is the cell executing a program it has always carried, a program that exists precisely for situations where the damage is too extensive, the repair too incomplete, the risk of proceeding too high. The cell activates a cascade of proteases called caspases. The nucleus condenses. The DNA is cleaved at internucleosomal sites. The cell breaks into membrane-bound fragments that neighboring cells and immune cells will clear away cleanly, without inflammation, without spillage. The cell ends, the damage ends with it. There is a third option, which is senescence. A permanent arrest without death, a state the cell enters when the damage is severe enough to halt division, but not severe enough or not the right kind to trigger apoptosis. Senescent cells persist. They remain metabolically active. They don't replicate. They accumulate, particularly with age, in tissues throughout the body. What they contribute to: chronic inflammation, tissue dysfunction, the general biological landscape of aging, is a subject researchers are still working through. But that comes later in this script. For now, the thing worth sitting with is this. The checkpoint exists because replication errors and unrepaired damage are not theoretical concerns. They happen. The checkpoint is not a failsafe against a rare event. It is a routine part of the cell cycle engaged regularly, because the conditions of being alive produce enough damage that a pause and check mechanism was, and is, genuinely necessary. The cell that stopped is not a broken cell. It is a cell doing exactly what it was built to do, in a situation the cell's biology anticipated, running a program that has existed in some form since before your lineage diverged from organisms that look nothing like you. The checkpoint is old. The problem it addresses is older. What happens next depends on what the repair systems find when they get there. Usually they find enough. The damage is cleared. The inhibition lifts. The cycle resumes. But not always. And in that gap, in the fraction of cases where the checkpoint is bypassed or the repair incomplete or the apoptosis signal absent, something begins that the body will spend considerable resources trying to contain. The cell continues when it shouldn't. That's all it takes, just that. There is a state a cell can enter to that has no clean analogy in the experience of being a person. It is not rest. It is not illness. It is not waiting in any sense that implies something is coming. A senescent cell is a cell that has stopped dividing and will not divide again. Not because it lacks the materials, not because it's damaged beyond function in the way a dying cell is damaged, but because something in its internal accounting has registered a threshold crossed, a signal accumulated beyond the point where continuing was the safer option, and it has responded by locking itself in place. Permanently. That word deserves its own line. Permanently, not paused, not arrested in the temporary way a checkpoint arrest is temporary. The senescent state, once fully established, is stable. The cell remains. It continues its metabolic work. It makes proteins. It maintains its membranes. It responds to signals from its environment. It simply does not replicate. The gene expression programs that would drive cell division are suppressed, and they stay suppressed, and the cell persists in that suppression for as long as it survives. The trigger for this can be several things. Telomere shortening, the progressive erosion of the chromosome end caps with each round of replication, is one of the most studied. When a telomere shortens past a critical length, the chromosome end loses the protective protein complex that shields it, and the exposed end resembles, to the cell's damage-sensing machinery, a double-strand break. The DNA damage response activates. The checkpoint proteins engage, and rather than repairing something that cannot be conventionally repaired, the cell transitions into senescence. Other triggers exist. Oncogene activation, the kind that happens in an early cancer cell when a growth-promoting gene is aberrantly switched on, can paradoxically drive cells into senescence rather than proliferation. The logic, evolutionarily, is something like a circuit breaker. Too much growth signal triggers a permanent halt rather than uncontrolled expansion. Oxidative stress, sufficient DNA damage, certain developmental signals, each can tip a cell into this state. What I find genuinely interesting, and not in a way that resolves cleanly, is that senescence is protective and costly at the same time, not sequentially, not protective first and then costly later, though there is some of that. Simultaneously, the same state. The protection is real. A senescent cell does not become a cancer cell. It has exited the proliferative pool. Whatever mutations it carries, whatever genomic instability it has accumulated, it will not replicate those mutations forward. In early cancer development, in tissues under high replicative stress, senescence is a genuine barrier. Mice engineered to have impaired senescence pathways develop cancers at higher rates. The pathway does what it appears to do. The cost is also real. Senescent cells secrete things. This is the part that took researchers time to fully appreciate, and it changed the way senescence was understood. A senescent cell is not silent. It is, in a specific and measurable way, loud. It releases a mixture of inflammatory cytokines, proteases, growth factors, and signaling molecules collectively called the senescence-associated secretory phenotype, SASP. The composition varies by cell type and trigger, but the general character of it is pro-inflammatory. The senescent cell is, in effect, sending out signals that alter the tissue environment around it. A cup of cold tea was sitting on the desk while I wrote this section. Forgotten, the way things get forgotten when you're deep in something. And I looked at it and thought about how long it had been there. In young tissue with few senescent cells, the SASP signals have roles. They attract immune cells that clear senescent cells from the tissue. They participate in wound healing. They contribute to developmental processes in the embryo. The secretory phenotype is not purely harmful in the context it evolved for, but senescent cells accumulate. With age, with cumulative damage, with the gradual shortening of telomeres across replicating populations, more cells reach the threshold. More cells enter and stay in the senescent state. The immune system, itself clears them less efficiently. They persist in tissues, in fat, in the vasculature, in the lung, in joints, and the SASP continues. Chronic low-grade inflammation, tissue stiffness, impaired regeneration as the stem cell niche is altered by senescent cell signaling. The degradative enzymes in the SASP remodel the extracellular matrix in ways that change tissue architecture over years. These are not acute events. They are slow, cumulative, distributed across an entire organism over decades. This is part of what aging is. Not all of it. Aging is more than senescence, more than any single cellular mechanism, but this is part of what it is. The body's own protection against cancer, accumulating past the point where it can be efficiently cleared, becoming a source of the slow dysfunction it was meant to prevent. There are researchers working on what are called senolytics, compounds that selectively induce apoptosis in senescent cells, clearing them from tissues. Some show effects in animal models. Some are in early clinical trials. The results are interesting. The biology is complicated. Whether clearing senescent cells will meaningfully alter human aging trajectories, and at what cost, and in which tissues, and when in the lifespan, these questions do not have clean answers yet. What exists tonight in your tissues are cells that stop dividing and stayed stopped. Quiet in one sense, secreting in another, doing what they do while the cells around them do what they do. Some of them have been senescent for years, some became senescent recently, some will be cleared by immune activity before they cause any lasting change to the tissue around them, some won't. And that's the thing about senescence that I keep sitting with. The way it refuses to be simply good or simply bad. It is both, depending on when and where and how many. It saved you probably from a cell division that would have compounded a problem, and it stays afterward in the place where it stopped, doing something the tissue didn't ask for, in a state that has no exit built into it. Still there, still secreting, still not dead, not damaged, not corrupted, lost deliberately by design in the way a margin exists on a printed page. The text doesn't reach the edge. Something is held back. These are telomeres, and what they are made of is repetitive, almost defiantly repetitive. The same 6-base sequence, TTAGGG, repeated in tandem thousands of times. In a young human cell, a telomere might span somewhere between 8 and 15 kilobases of this sequence. All of it redundant. All of it in a strict informational sense saying nothing, just the same 6 letters over and over, capping the chromosome end in a structure that, along with the proteins bound to it, keeps the DNA repair machinery from treating the chromosome tip as a break that needs fixing, which is what it would otherwise look like. A chromosome end structurally resembles a double-strand break. The DNA just ends. There is no template continuing on the other side, without something marking it as a legitimate terminus rather than damage. The cell's repair systems would attempt to fix it. They would fuse chromosome ends together. They would trigger damage responses. The result would be chromosomal rearrangements, catastrophic genomic instability, the kind of structural chaos that ends cells. The telomere prevents this. The proteins that bind to it, a complex called shelterin, composed of 6 subunits with specific and non-redundant roles, organize the telomeric DNA into a structure called a T-loop, where the single-stranded overhang at the very end folds back and tucks into the double-stranded region behind it. The end is hidden. The repair machinery passes over it. The chromosome is safe, until it isn't. Something was dripping outside, distant and irregular, the way plumbing sounds in the walls of a building at night when no one is using anything. Every time a cell divides and its DNA is replicated, the telomere gets shorter. Not because the replication machinery fails, it doesn't fail exactly, but because of something called the end replication problem. DNA polymerase can only synthesize in one direction, and it requires a shorter RNA primer to begin. At the very end of a linear chromosome, when the primer is removed after replication, the gap it leaves cannot be filled. There is no upstream template. A small stretch of sequence is lost with each division. How small depends on cell type and conditions. Somewhere between 50 and 200 base pairs per division is a commonly cited range. Against a starting telomere of 10,000 base pairs, that seems manageable. Over 50 cell divisions, it starts to matter. Over 100, the telomere is critically short. This has been true for every cell in your body that has ever divided. The shortening is not a malfunction. It was present in the biology of organisms hundreds of millions of years before anything resembling a human existed. The end replication problem is an intrinsic feature of linear chromosomes and DNA polymerase, which means every organism with linear chromosomes has had to either solve it or accept its consequences. Germline cells and stem cells in certain tissues express telomerase, an enzyme that adds T, TA, G, repeats back to chromosome ends, partially compensating for the loss. Most somatic cells, the ordinary working cells of the body, express very little of it. Their telomeres shorten, steadily, over a lifetime. When a telomere shortens past the point where the sheltering complex can maintain the T-loop structure, when the buffer zone has been consumed down to its functional minimum, the chromosome end becomes exposed, not dramatically, not all at once, but exposed enough that the DNA damage response begins to recognize it. ATM and ATR kinases activate. The checkpoint signals go out. The cell cycle arrests. This is replicative senescence. The cell stops dividing not because of any acute damage, not because of a double-strand break or an oxidized base or a UV-induced dimer, but because its telomeres have reached the end of what they had to give. The margin has been used up, the chromosome end is visible, and the cell sits with that. There is something about the telomere mechanism that I keep coming back to, not as metaphor, because I think the biology is strange enough without borrowing from outside it, but as design. The shortening is built in. The limit is built in. Every division a somatic cell makes is, in a sense, drawing down from a finite reserve. Not unlimitedly, not carelessly, but against something that will eventually run out. The evolutionary logic, as best it's been reconstructed, has to do with cancer suppression. Unlimited replication is what cancer is, a mechanism that imposes a replicative ceiling that makes endless division structurally impossible for most cells, is a mechanism that reduces malignant transformation. The cost is cellular aging. The cost is tissue decline. The cost is that the cells which have divided the most are the ones closest to the end of their capacity. Whether that trade was worth it is not a question evolution answered. It is a question it avoided. The trade produced organisms that survived long enough to reproduce, and that was sufficient. Your telomeres right now are shorter than they were when you were born. Not catastrophically, not even measurably in any way you'd notice, but shorter. Each tissue at its own rate, each cell type according to how often it is divided. Each chromosome end carrying a record of how many times that particular lineage has replicated since the beginning. The T-to-A-G-G-G repeats are still there. For now, the sheltering complex is holding the T-loop closed. The chromosome ends are hidden from the machinery that would otherwise act on them. The margin is still wide enough. For now, the ends are protected. They are being consumed, and they are enough. Most cells accept the limit. They divide as many times as their telomeres allow, dozens, sometimes more than 100 for certain cell types, and then they stop. They enter senescence, or they die, and the tissue around them continues with whatever replicative capacity remains in younger cells or in stem cell populations that still maintain some telomerase activity. The limit is not catastrophic. It is, for most cells, simply the end of their particular run. Cancer cells are different, not because they are stronger exactly, not because they have acquired some additional capacity that normal cells lack. In the majority of cases, somewhere around 85 to 90% of human cancers, what they have done is reactivate something that was already there, already encoded, already present in the genome, telomerase. Telomerase is a ribonucleoprotein enzyme. It has two core components, a reverse transcriptase called TERT, which does the enzymatic work, and an RNA template called TERC, which carries the sequence CCAAUG, complementary to the TTAG-G telomere repeat that TERT uses to extend chromosome ends. The enzyme adds telomeric repeats back to the 3' overhang. It rebuilds what replication has consumed. In germline cells, in certain stem cells, it runs continuously enough to maintain telomere length across divisions. In most somatic cells, it is silenced, transcriptionally repressed in ways that have been selected across evolutionary time, specifically because unlimited somatic replication is dangerous. Cancer cells turn it back on. The mechanisms vary. Mutations in the TERT promoter are among the most common, found in melanoma, bladder cancer, glioblastoma, hepatocellular carcinoma, among others. Some cancers amplify the TERT gene. Some use epigenetic changes to alter its regulation. The specific route differs by cancer type. The outcome is the same. Telomerase becomes active in cells that should not have it, and the telomeres that would otherwise shorten to the point of crisis are maintained, or in some cases lengthened. A window had been left open a crack somewhere nearby, just enough that the air in the room had changed slightly, taken on a faint coolness that hadn't been there an hour ago. This is the part that takes a moment to sit with properly. The telomere shortening mechanism, the one that limits somatic cell division, that enforces replicative senescence, that acts as a barrier to malignant expansion, is itself a cancer suppression system. It imposes a ceiling. Cancer circumvents the ceiling not by smashing through it, but by removing it. By restoring the enzyme that was deliberately silenced, in most cells, for this exact reason. The body built a wall. The cell found the gene for a gate. I was going to use that as a comparison, and I'm not sure it holds, so I'll leave it there and move on. What telomerase reactivation means practically is that a cancer cell that would otherwise reach its replicative limit, that would otherwise trigger a checkpoint or enter senescence, or reach the chromosomal instability of telomere crisis, continues dividing indefinitely. The telomere clock, which runs in every normal somatic cell from birth, is stopped or reversed. The cell is no longer drawing down from a finite reserve. It has made itself, in this one specific sense, unlimited. The remaining 15% of cancers that don't use telomerase reactivation use something called AL. T. Alternative lengthening of telomeres. A recombination-based mechanism. The cell uses homologous recombination to copy telomeric sequences from one chromosome to another, extending the ends without telomerase. It is less well understood. It appears to produce telomeres that are more variable in length, less stably maintained, but sufficient for continued replication. Two different solutions to the same problem. Both arrived at independently in different cancer lineages, because any cancer cell that failed to solve the telomere problem eventually stopped dividing, and any cancer cell that solved it did not. The evolutionary logic inside a tumor is not subtle. Here is the irony that I find hardest to hold cleanly. The germline cells in your body, the cells that produce eggs and sperm, the cells that carry your genetic information forward into another generation, maintain telomeres through telomerase. They are, in this specific respect, doing the same thing cancer cells do. Running the same enzyme, solving the same end replication problem. The difference is regulation, where, when, how much, in which cellular context. But the molecule itself is not inherently malignant. Telomerase is a tool. The germline uses it carefully in context for the purpose of hereditary continuity. Cancer uses it without restraint, without context, in service of its own expansion. The same enzyme. Researchers have been interested in telomerase as a cancer target for decades. If telomerase is what allows cancer cells to divide indefinitely, then inhibiting telomerase should, in principle, restore the replicative limit. In practice, the picture is more complicated. There is a lag between telomerase inhibition and telomere shortening, and the lag is long enough that cells accumulate new mutations during it. There are toxicity concerns involving normal telomerase-expressing tissues. There is the ALT pathway, which doesn't use telomerase at all. The research continues. What is sitting in your own tissues right now, in the cells that are dividing, in the telomeres that are shortening, in the rare cases where a cell has begun the process of reactivating something it should have kept silent, is not something you can feel, not something that is, tonight, in any acute sense, a problem. Most cells accept the limit, most always will, some over a lifetime don't. And those ones carry an ancient enzyme in a new context, using it for something the body built it to prevent. In a quiet expansion that began, in every cancer that has ever existed, with a single cell that found a way to keep going. For most of the 20th century, the working assumption in molecular biology was that DNA was essentially still, indestructible. Researchers knew UV light caused damage, knew certain chemicals were mutagenic, but the molecule itself, in the ordinary conditions of a living cell, was thought to be largely inert between assaults. It sat there. It held its sequence. The problem of mutation was mostly framed as a problem of external insult, not internal chemistry. Thomas Lindahl didn't begin his career intending to challenge this. He was working in the early 1970s on RNA degradation, a different molecule, a known problem, nothing controversial. RNA is unstable, everyone knew that. And Lindahl was studying the chemistry of why. And in that work, thinking about the hydrolytic reactions that break down RNA, he began to wonder about DNA. Specifically, he began to calculate, back of the envelope, then more rigorously, how stable DNA actually was under physiological conditions, not under radiation, not under mutagenic exposure, just sitting inside a warm, wet cell at body temperature, surrounded by water. The number he arrived at was uncomfortable. Spontaneous hydrolysis alone, depurination, deamination, the ordinary reactivity of DNA with the water it was bathed in, was occurring at a rate high enough that a genome, if left unrepaired over the course of a day, would accumulate damage incompatible with cellular function. The sequence could not survive its own chemistry without something actively maintaining it. This was not the consensus view. The consensus view was that DNA was the stable archive, the reliable record, the molecule that could be trusted to hold its information across generations. Lindahl's calculation suggested that this stability was not intrinsic. It was maintained, actively, continuously, by enzymatic systems that nobody had fully looked for because nobody had fully suspected they were necessary. He went looking for them anyway. Something had changed in the room while I was writing this section. The quality of the quiet, the particular stillness that settles around 2 a.m, when even the building stops making sounds. What Lindahl found, beginning with the identification of uracil A glycosylase in 1974, was the first direct evidence for active base excision repair. The enzyme he isolated could remove uracil from DNA, the base that forms when cytosine spontaneously deaminates, which happens, he calculated, roughly 100 times per cell per day. If no one was removing it, and if it went uncorrected through replication, the mutation rate in living organisms would be orders of magnitude higher than what was actually observed. The enzyme was the explanation. Its existence confirmed the problem. The two things, the rate of spontaneous damage and the existence of a repair system, fit together in a way that made the old assumption of passive stability difficult to maintain. The field did not immediately reorganize around this. Scientific consensus doesn't tend to do that. Papers get published. Some are received with interest and some with skepticism. Competing frameworks persist longer than the evidence perhaps justifies. What Lindahl had found was genuinely important, and it was recognized as interesting, but the full weight of it, the understanding that DNA stability was a product of continuous active repair rather than intrinsic chemical inertness, took time to become the foundational assumption it is now. He kept working. Through the 1980s and 1990s, his lab and others mapped the base excision repair pathway in increasing detail. The glycosylases, the AP endonucleases, the polymerases and ligases that completed the repair. Each component identified added another piece to a picture that was, by the time it was complete, considerably more elaborate than the early calculations had suggested. The repair wasn't a single enzyme doing a single job, it was a layered, redundant, evolutionarily ancient system with specialized components for different types of damage. Paul Modrich was working in parallel on mismatch repair, the pathway that corrects replication errors. Aziz Sankar was characterizing nucleotide excision repair, the pathway that removes bulky lesions like thymine dimers. 3 researchers, 3 pathways, 3 decades of careful biochemical work converging on the same fundamental understanding. DNA is under continuous chemical assault, and life depends on systems that are equally continuous in their response to it. The Nobel Prize in Chemistry arrived in 2015. Lindahl was in his late 70s by then. The work that formed the foundation of the prize had been done 40 years earlier, in a period when the question he was asking wasn't yet considered central. That gap between doing the work and being recognized for it in the largest formal way available says something about how slowly the center of a field moves, even when the evidence at the edges has been accumulating for decades. I'm not sure what to do with that exactly. It's not a complaint, it's just how it goes, and Lindahl continued working throughout, which is its own kind of answer. What his work gave us, what it gave you, in the sense that understanding your own biology is something you can now do more accurately, is a corrected picture. DNA is not a stable archive that occasionally gets damaged. It is an unstable molecule that is continuously damaged and continuously repaired, and the stability we observe is the net result of those two processes running simultaneously, every second, in every cell. Right now, while your breathing has slowed and the room is whatever temperature it is, and your body is doing what bodies do in the hours after midnight, the repair is running. It was running before Lindahl calculated why it had to be. It was running before anyone knew to look for it. It will keep running regardless of whether you ever think about it again. The stability you have is not given. It is made. Every day. Not every part of your genome receives the same attention. This is something that took me a while to fully absorb, not the fact of it, which is fairly straightforward once you encounter it, but the implications. We tend to think of the genome as the unified thing, a single document maintained under consistent conditions, every base pair equally subject to the same repair processes. But that's not quite right. The genome is more like a city than a document, if that comparison holds at all, and I'm not sure it does, so I'll use it once and let it go. Some neighborhoods are continuously active, well lit, frequently traversed. Others are quiet, dark, rarely visited. The maintenance is not the same in both. The mechanism responsible for this difference is called transcription-coupled nucleotide excision repair, TCNER. It is a sub-pathway of the nucleotide excision repair system described in an earlier chapter, the one that removes bulky lesions, thymine dimers, distortions that kink the helix, and require a stretch of DNA to be cut out and resynthesized. Global Genomic NER, the general form of this pathway, scans the entire genome for these distortions. It finds them, eventually. But eventually is a relative term, and in a genome 3 billion bases long, with damage accumulating continuously, the time between a lesion forming and being found can vary considerably depending on where in the genome it sits. TCNER is faster. Specifically faster for genes that are being actively transcribed. Here is why, and the mechanism is stranger and more elegant than the name suggests. When RNA polymerase, the enzyme that reads a DNA template and synthesizes the corresponding RNA, encounters a bulky lesion in the template strand, it stalls. It cannot read through a thymine dimer or a large chemical adduct. The helix distortion prevents it from advancing, so it stops, sitting on the DNA at the site of damage, neither completing the transcript nor releasing the template. In most cellular contexts, a stalled enzyme is a problem. Here, it becomes something else. The stalled RNA polymerase is the signal. Its presence, its specific geometry of stalling, the way it sits on the damaged template with a partially synthesized RNA attached, recruits repair factors. CSB protein arrives first, interacting with the stalled polymerase and remodeling the complex. CSA follows, part of a ubiquitin ligase complex that modifies nearby proteins and opens the chromatin. TFIIH is recruited, the same multi-subunit complex involved in global NER, the one that contains the helicases XPB and XPD that unwind the helix around the damage. From there, the pathway converges with global NER, dual incision, gap-filling, ligation. The lesion is removed. The polymerase is displaced or allowed to backtrack. Transcription can resume. What this means is that a gene currently being read, a gene whose RNA polymerases are actively moving along its template strand, is under continuous surveillance by a repair mechanism that global NER cannot match for speed. Every polymerase that stalls at a lesion immediately flags the site for repair. The active gene is not just being expressed, it is being monitored. The rain had started at some point without my noticing, and I only registered it when a gust pushed it briefly against the glass. Genes that are ahisence, not currently being transcribed, not active in this cell type or at this point in development, receive no such benefit. They rely on global NER, which does its work but does it more slowly, scanning the genome at a pace determined by the abundance of damage recognition proteins and the sheer length of the sequence to be surveilled. Damage in a silent gene may persist longer before being found, longer before being cleared. Whether that matters depends on what the gene is and what the damage does when a replication fork eventually passes through. For a gene that will never be expressed in this cell, the consequences may be limited. For a gene that becomes active later, switched on by a developmental signal, or by a cellular stress response, or by a change in an environment, an unrepaired lesion inherited through replication may carry forward into function. There is something pragmatic about TCNER that I find more interesting than if it were purely elegant. It didn't evolve because the cell decided to prioritize active genes. It evolved because RNA polymerase was already there, already stalling at lesions, already creating a physical signal that existing repair proteins could recognize and respond to. The coupling between transcription and repair is, in a sense, opportunistic. The transcription machinery was repurposed, at least partially, as a damage sensor. Not because it was designed for that, because it was present, and its behavior at damaged sites was informative, and the proteins that responded to that information survived better. Evolution working with what already existed, which is what it always does. Your genome right now has regions that are being actively read, genes expressed in the cells currently running their maintenance programs, their metabolic work, their signaling conversations across tissue. Those regions in the template strands of those active genes are under tighter surveillance than the rest. Damage that forms there tonight is more likely to be found quickly, more likely to be cleared before anything downstream depends on the sequence. The rest of the genome waits for the global scan, patient, unmonitored, accumulating what it accumulates. Most of it doesn't matter. Some of it, in which genes and in which cells and with what eventual consequence, is something the biology of each body works out on its own timeline, in its own way, without asking. You are already in it. Whatever hour this is, whatever degree of almost sleep your body has reached, you're already in the biological state this chapter is about. Not metaphorically, your core temperature has dropped a fraction of a degree since you lay down. Certain hormone concentrations have shifted. The activity patterns in your brain have changed in ways that no act of will could replicate. Your body has moved without asking you into a different mode of operation, and the repair systems have noticed. There is a structure in the hypothalamus called the suprachiasmatic nucleus, a small cluster of around 20,000 neurons sitting just above the optic chiasm, receiving light information from the retina and using it to synchronize a molecular clock that runs in nearly every cell in your body. The SCN is the master pacemaker, but the clocks it synchronizes are distributed, present in your liver cells, your skin cells, your gut epithelium, your immune cells, each one running the same basic molecular feedback loop. Clock and BMAL1 proteins driving expression of PER and CRY proteins, which accumulate, inhibit clock and BMAL1, degrade, and allow the cycle to begin again. One cycle every 24 hours, give or take. These clocks control gene expression. broadly, pervasively in ways researchers are still mapping. Which proteins get made, and in what quantities and at what times, much of this is under circadian control, including, it turns out, a number of proteins involved in DNA repair. The specifics are not uniform across repair pathways or cell types. The picture is genuinely complex and still being assembled. But the general finding, supported by a growing body of work in rodent models and increasingly in human tissue, is that certain repair gene expression peaks at specific circadian phases. Nucleotide excision repair components show circadian oscillation. Base excision repair enzymes show it. The timing varies by tissue. The principle, that repair capacity is not flat across the 24-hour cycle but rises and falls with the molecular clock, appears to hold broadly. What this means is that there are hours during which your cells are better equipped to handle certain types of damage than others. Not infinitely better, not so dramatically better that it's a simple on-off switch, but measurably different. The repair machinery is expressed at higher levels at some phases than others, and the phase that aligns with sleep, with darkness, with temperature drop, with the hormonal milieu of nighttime, is for many repair systems closer to peak than trough. Sleep is a condition. That is the way I find most useful to think about it here. Not as rest, not as downtime, not as the absence of waking, but as a specific physiological condition that certain biological processes require. Some repair works under any condition. Some works better when the circadian clock is aligned with the repair gene's expression peak. Some works better when the replicative load is lower, when fewer cells are dividing, and the repair machinery has less competition for its substrates and cofactors. During sleep, in tissues with high turnover, the gut epithelium, certain immune populations, cell division slows. Not everywhere and not completely. But the replicative rate in some tissues drops measurably in the sleep phase, and that drop changes the landscape. Fewer replication forks running means fewer forks encountering unrepaired lesions. Less competition between replication and repair for the same stretch of DNA. A slight but real shift in the balance between damage accumulation and damage resolution. The weight of the blanket is something you probably stopped noticing an hour ago. The way the body accommodates pressure that doesn't change. There is work on what happens when sleep is disrupted chronically. Not a single bad night, one night of poor sleep does not produce detectable genomic instability in any study I've found cited with confidence. But sustained disruption, shift work, chronic circadian misalignment. The data in animal models is fairly consistent. Disrupted circadian rhythms correlate with reduced repair gene expression at expected phases, with higher rates of DNA damage accumulation in certain tissues, with increased genomic instability over time. The mechanism is not fully resolved. It is likely several mechanisms operating together, but the correlation is real, and the proposed pathway through circadian regulation of repair gene expression is plausible and supported. In human epidemiology, shift workers show elevated cancer rates in several tissue types. The International Agency for Research on Cancer classified shift work involving circadian disruption as a probable human carcinogen in 2007. Probable, not certain. The epidemiology is complicated by confounding factors, but the biological mechanism underlying the association that circadian disruption impairs DNA repair capacity in phase-dependent ways, allowing damage to accumulate that would otherwise be cleared, is no longer speculative. It is at this point a reasonable thing to believe, which means that what you are doing right now, lying in the dark at the hour your body expects darkness, with your core temperature dropping and your SCN no longer receiving strong photic input, and your repair gene expression somewhere near its daily peak, is not nothing. It is not simply the absence of waking, it is a condition, one that the repair systems running in every cell of your body are, in ways that are measurable and real, better suited to work within. Your stillness is doing something, not because you decided it, not because you're particularly still or particularly asleep or doing this especially well, just because this is the hour, and your body is in the state it evolved to be in at this hour, and the repair is running in the conditions it runs best in. Something in the rate of correction is higher right now than it was this afternoon. Some lesion that formed today is being found tonight. Some strand that was nicked is already whole. You didn't do anything, you just stayed. Start with one cell in one tissue on one day. The damage it sustains, depurination, oxidative lesions, deamination events, the occasional replication error that slips past proofreading, adds up to somewhere between 10,000 and 1 million molecular lesions. The range is wide because the sources vary, because the tissue type matters, because oxidative stress fluctuates and replication rates differ, and UV exposure depends on where the cell lives in the body and what you did that day. Call it, conservatively, 100,000 lesions. In one cell today, almost all of them are repaired. The glycosylases find the wrong bases and pull them out. The AP endonucleases nick the backbone at a basic sites. The polymerases fill the gaps. The ligases seal the nicks. Nucleotide excision repair finds the bulky distortions and cuts them out in long stretches and resynthesizes what was removed. Mismatch repair scans the newly replicated DNA and corrects the misincorporations that proofreading missed. Double-strand breaks are held by Q searched by RAD51, filled in by homologous recombination or rejoined by NHEJ. The system runs. The lesions are cleared. Most of them, the error rate of DNA replication after all repair and proofreading, is approximately 1 mistake per billion bases. In a genome of 3 billion base pairs, that means each cell division produces roughly 3 new mutations. Some studies suggest fewer. Some suggest slightly more. 3 is a reasonable working figure. 3 per division. In tissues that divide frequently, colon epithelium, skin, bone marrow, cells divide many times over a lifetime. Estimates for colorectal stem cells suggest somewhere around 40 divisions per year. Over a 70-year life, that's roughly 2,800 divisions. At 3 mutations per division, a single colorectal stem cell lineage accumulates in the range of 8,000 mutations over a lifetime. 8,000 in one cell lineage. Most of them are in intergenic regions, stretches of the genome between genes, in regulatory sequences that aren't critical, in repetitive elements that can absorb a changed letter without any functional consequence. The genome is large, and the regions where a mutation meaningfully changes a protein or a regulatory element are a small fraction of the whole. Most mutations do nothing. Your breathing has probably shifted in the last few minutes, slower, more irregular, the kind that happens at the edge of sleep when the body is making decisions the conscious mind no longer participates in. But some mutations land somewhere that matters. A changed base in a tumor suppressor gene, a frame shift in a DNA repair gene itself. The dark irony of that. A mutation that impairs the system preventing further mutation. A single nucleotide change in a regulatory element that subtly alters the expression of a growth-controlling gene. These are rare as a fraction of all mutations. As an absolute number accumulated across 30 trillion cells over decades, they are not rare at all. They are inevitable. Not in any specific cell, in any specific gene, on any specific day. That is genuinely unpredictable. But in aggregate, across a lifetime of dividing cells, the probability that some mutation lands somewhere consequential approaches certainty. This is not pessimism, it is arithmetic. what the body has against this arithmetic is surveillance. Checkpoint systems that catch the cells behaving abnormally. Immune monitoring that recognizes and eliminates cells displaying markers of transformation. Apoptosis programs that activate when damage crosses certain thresholds. Senescence that freezes cells that shouldn't continue. These systems catch most of what the repair systems missed. Most. The word keeps appearing in this script because it keeps being the honest word. Not all, not infallible, most. And in the gap between most and all, in the small percentage of mutations that escape repair, and the smaller percentage of those that land in functional regions, and the smaller percentage still that alter function in ways that matter, and the even smaller percentage that survive immune surveillance and checkpoint control and accumulate further in daughter cells. In that compounded small percentage, cancer begins. Aging accumulates. The occasional inherited mutation present from fertilization alters the probability distribution for everything that follows. These are not failures of the system, they are the residue of a system running at scale. The output of 30 trillion cells sustaining damage and repairing it every day for decades, at an error rate of 1 in a billion per base copied. The numbers are so large that even an extraordinarily low error rate produces thousands of mutations per cell lineage. The wonder is not that some slip through. The wonder is that the error rate is as low as it is. Go back to the single cell now. One cell somewhere in your body, currently in whatever phase of its cycle it inhabits tonight. It has repaired something in the last hour, several things probably. The lesions were found and the sequences were restored and the strands were sealed and the cell continued, carrying a genome that is nearly identical to what it started with. Nearly. Somewhere in its 3 billion bases, one letter is different from what it was a year ago. Or 5 are. Or 20. The cell does not know. The proteins it makes from those sequences are probably identical or close enough that nothing changes. The cell divides eventually and passes those letters forward, and the daughter cells carry them. 1 cell in 30 trillion. On one night of the several thousand, your body will be alive. Already whole again. For now, running in the dark, with its corrected genome and its irreducible residue of change. Doing what it has always done without being asked. You didn't ask for any of this. Not the damage, not the repair, not the 37 trillion cell infrastructure running beneath the threshold of anything you've ever felt. None of it was requested. None of it requires your attention or your gratitude or your awareness to function. The glycosylases were finding wrong bases in your DNA, long before you knew what a glycosylase was. RAD51 was searching your genome for matching sequences when you were an infant, when you were a fetus, when you were a cluster of cells that had not yet decided what kind of organism it was going to become. The repair is older than you, older than your species, older than your genus, your family, your order. The base excision repair pathway, the one that removes oxidized and deaminated bases from DNA, has homologs in bacteria. The proteins that carry it out have been recognizable across more than 2 billion years of life. What is running in your cells tonight, finding a basic sites and filling gaps and sealing nicks in the dark, is something your ancestry has been passing forward since before there were animals, before there were multicellular organisms of any kind. The repair was there, in the first cells that had DNA worth protecting, and every organism between those cells, and you either kept it or died. You kept it, not through any decision, not through fitness in any sense you chose. You kept it the way all inheritance works, passively, automatically, through the fact of being here at all. Every repair enzyme in your body arrived via an unbroken chain of organisms that survived long enough to reproduce, and every one of those organisms survived in part because the repair worked. The chain is 4 billion years long. It has not broken once. That is not metaphor. That is the actual record. I find this genuinely difficult to hold in mind for more than a few seconds without it slipping back into abstraction. There's a gap between knowing something intellectually and feeling its full weight, and the full weight of 4 billion years of unbroken cellular inheritance is not something the human mind is built to sustain. But for a moment, just a moment, in the dark, in the specific quiet that exists right now wherever you are, it's worth trying. Something in the temperature of the room has settled, the kind of settling that happens in the small hours when the building stops its thermal adjustments and everything just sits. The repair running in your cells right now has no purpose in any intentional sense. There is no goal. The enzymes don't know what they're protecting or why it matters. Mutagenesis doesn't understand that the mismatch it found could, uncorrected, contribute to a mutation in a tumor suppressor gene. OGG1 doesn't know that the 8-oxyguanine it removed was one of 10,000 formed today by the ordinary exhaust of mitochondrial respiration. The checkpoint kinase that halted a cell cycle doesn't know what it prevented. The ligation that sealed a strand doesn't know what the strand contains. They just do what they do. What they've always done. What they were shaped over incomprehensible time to do. And you are, in the most literal biological sense, the outcome of that doing. Not a metaphorical outcome, not a philosophical one. Your actual physical existence, the fact that your cells have not accumulated catastrophic genomic instability, that your tissues function, that you are lying somewhere right now breathing in the way you're breathing, is the outcome of repair systems running correctly more often than not, across every cell division you have ever undergone, from the first mitotic division of the zygote that became you to the divisions happening in your bone marrow and your gut lining tonight. Most of them worked. Most. And here we are. If this script has given you something, a few hours of quiet, a sense of what's running beneath the surface of your skin, a reason to feel something other than indifferent toward your own biology, then it did what it was made to do. If you want to help keep this going, the most honest thing I can say is, a like on this video costs you nothing and genuinely helps other people find it. Subscribing means the next time I finish something, it finds its way to you. If you've been watching for a while and feel something like affiliation with this space, there's a membership option that keeps the channel running in a more direct way. I think about those people more than they know, and if something in this script made you want to say something, the comments are there. I read them. Slowly, and not always promptly, but genuinely, and that's all of it. The repair doesn't need anything from you tonight. It's already working. The strand that broke while you were listening to this, somewhere in a skin cell, in a gut cell, in a lymphocyte circulating through tissue you'll never feel, has already been found. The ends were held. The search ran through the genome and came back with the right sequence. The bases were laid in. The nick was sealed. The cell continued. Somewhere in your body right now, in the dark, at whatever depth of almost sleep you've reached, a ligase just finished its work. A strand is whole. You didn't feel it. You didn't need to.